Welcome, everyone. Today we're covering the histology of the nervous system, focusing on microscopic structure and cell types that make neural communication possible. By the end of this lecture, you should be able to identify major nervous tissue components, such as neurons, glial cells, and peripheral nerve structures, and differentiate central from peripheral nervous system elements. The nervous system consists of two main categories of cells, neurons and glial cells. Neurons are responsible for electrical signaling, while glial cells support and protect neurons. Both are essential, as neurons cannot function without glial support. Neurons are the functional unit of the nervous system and typically are post-mitotic, meaning that they do not divide, while glial cells, by contrast, are capable of mitosis and provide physical, metabolic, and insulating support, which is why most central nervous system tumors arise from glial cells rather than neurons. Neurons have some distinct structures histologically, such as Nissl bodies. These are clumps of rough endoplasmic reticulum as well as ribosomes, and these reflect high protein synthesis for neurotransmitter production. And a neuropil, which is the meshwork of dendrites and axons that fills the space between the neuron cell bodies. This would be the neuron cell body here, with its projections coming off the side here, here, and here. These structural features allow for neurons to receive, integrate, and send information. One of our first glial cells is a satellite cell. These are glial cells in the peripheral nervous system, so outside of the brain and spinal cord, and they surround neuron cell bodies and ganglia, or not, they surround neuron cell bodies inside of the ganglia, and they help to regulate the neuron environment. They are really focusing on supporting nutrient and waste exchange, and they function very similarly to astrocytes, which are located in the central nervous system. Schwann cells also belong to the peripheral nervous system, and each Schwann cell myelinates one axonal segment. The myelin sheath consists of concentric layers of cell membrane that act as a electrical insulator, increasing signal speed along axons. And this slide is showing how Schwann cells wrap around a single axon to form the myelin sheath. The myelin sheath is kind of this outer layer. And the more layers of myelin that are present, the faster the conduction velocity. And you can see these little myelin sheaths surrounding these axons here. Speaking of myelin, nodes of Ranvier are these gaps between Schwann cells where the axonal membrane is exposed. And this is where a saltatory conduction can take place. The electrical impulse can jump from node to node, which greatly increases conduction speed. Not every fiber is myelinated, though. Some fibers are unmyelinated, and unmyelinated fibers are still associated with Schwann cells, but they lack a or a multilayered myelin sheath. And multiple unmyelinated axons may sit near a single Schwann cell groove, and these fibers, though, will send signals much more slowly. And this is showing a comparison of a myelinated over here and a non-myelinated over here axon. So in myelinated fibers, Schwann cells wrap tightly and repeatedly around one axon. And in unmyelinated fibers, Schwann cells support multiple axons without forming true myelin. The key distinction is the presence or absence of a multi-layered membrane insulation. Moving into the central nervous system, oligodendrocytes are the central nervous system equivalent of Schwann cells, but with an important distinction. One oligodendrocyte, as seen here, can provide myelination to multiple axons, and this is why demyelination diseases like MS have widespread neurologic symptoms, because they are targeting these oligodendrocytes. Astrocytes, as I said, are like satellite cells in the peripheral nervous system. So astrocytes in the central nervous system are the most abundant CNS glial cell. They have foot processes seen here or here. And these foot processes help to form our blood-brain barrier. This regulates the entry of substances from the blood into neural tissue. They also contain glial fibrillary acidic protein or GFAP, which is a protein that's useful for immunostaining astrocytes in pathology. Microglial cells are the resident immune cells of the central nervous system. They become active during injury or infection and are capable of phagocytosis, which makes them essential for central nervous system immune defense as well as tissue cleanup. And lastly, ependymal cells line the ventricles and central canal of the spinal cord. They can contain cilia and microvilli and contribute to cerebral spinal fluid movement as well as absorption. In the choroid plexus, there are modified ependymal cells and they help to produce cerebral spinal fluid. This is showing kind of a breakdown of the different types of cells in their importance. So kind of looking at neurons and then looking at glial cells and a compare and contrasting of them both.